Cell cycle and cell division. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organism. Growth and reproduction are characteristic of cell, indeed of all living organism. All cell reproduce by dividing into two with each parallel cell giving rise to two daughter cell each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by a growth and division of a single parental cell and its progeny. In other words, such cycle of growth and division allow a single cell to form a structure consisting of millions of cells. Cell cycle. Cell division is a very important process in all living organisms. During the division of cell, DNA replication and cell growth also take place. All these processes, cell division, DNA replication and cell growth, hence have to take place in a coordinate way to ensure correct division and formation to progeny cell containing intact genome. The sequence of events by which a cell duplicates its genome, synthesis the other constituents of the cell and eventually divides into two daughter cells is termed cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosome DNA, the distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Phase of cell cycle. A typical eukaryotic cell cycle is illustrated by human cell in culture. This cell divided once in approximately even 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases, interphase and M phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represented the phase between two successive M phases. It is significant to note that in the 24 hours average duration of cell cycle of a human cell, cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosome karyokinesis and usually ends with division of cytoplasm cytokinesis. The interphase through called the resting phase in the time during which the cell is preparing for division. By undergoing both cell growth and DNA replication in an orderly manner, the interphase is divided into further phase, G phase, S phase, G2 phase. A diagrammatic view of cell cycle: cytokinesis, telophase, anaphase, metaphase, prophase, G2, S, G1. G1 phase corresponding go to the interval between mitosis and initiated of DNA replication. During G1 phase, the cell is metabolically active and continuously grows about does not replicate its DNA. S or synthesize phase marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increases in the chromosome number. If the cell had diploid or 2N number of chromosome at G1, even after S phase, the number of chromosomes remain the same, 2N. In animal cell during the S phase, during replication, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues.